found that this file was a little too big, so I divided it into 2 chapters, 2 parts. Might make it a little bit easier for you to upload to. So we're going to go to the reticular formation, which is also called the RAS, and its functioning. The RAS is reticular activating system. What it does is it sends impulses to the cerebral cortex to keep it conscious and alert, and it filters out repetitive, familiar, or weak stimuli. So you usually block out about 99% of everything that's going on around you. Now, this center that wakes up the brain is inhibited by sleep centers, alcohol, and drugs. So these are things that can actually shut down this ability to wake up the brain. If you have a severe injury to this part of the brain, it could result in permanent unconsciousness or coma. This is a look at where this reticular activating system radiates from. It goes up through the brainstem to the thalamus, which is the relay station in the diencephalon, and radiates that information to the cerebral cortex. It also takes in visual impulses, so we are wired to be awake when it's daylight, so the increase in light will stimulate the eyes to connect into this, the visual pathway to connect into this pathway as well. Language. We talked about Broca's area a little bit, but just to elaborate on this a little bit more, Broca's area is our motor area for speech, and Wernicke's is understanding language. These areas are usually dominant on the left-hand side of the brain, so the left hemisphere is usually where they're dominant, so the ability to speak, understand language comes in that area. There are Broca's and Wernicke's in the right hemisphere, but these have more of an emotional connection to speech. So with Broca's area, if you have damage to the right hemisphere, your speech may be flat, no emotional inflection at all in speech. Wernicke's damage to the right, a person may not be able to understand a person's intent when they're speaking in the emotional context of their speech, and they may not be able to understand if a person's telling a joke, that type of thing. These are the coverings of the brain, or what we call the meninges. These structures represent a double layer of meninge, and these are called a falx. Now you have a falx that goes in between the 2 cerebral hemispheres, and this is called the falx cerebri. You also have another double layer of meninge that goes in between the cerebrum and the cerebellum, and that's called the tentorium cerebelli. And then you also have a falx that goes in between the 2 hemispheres of the cerebellum, and that's called the falx cerebellum. So a falx is a double layer of meninge. This is the circulation of cerebral spinal fluid through the entire circulation system, through the ventricles and then through the subarachnoid space. You can see the arrows show you the direction of flow. And cerebral spinal fluid originates, it starts off being produced from the choroid plexuses. Here's the lateral ventricles that are found in the cerebrum, the 3rd ventricle in the diencephalon, and then you have the 4th ventricle located between the cerebellum and the pons. So that's where cerebral spinal fluid is produced. You produce about 500 milliliters per day. There's 150 milliliters in circulation at any given point in time. So you're actually replacing your cerebral spinal fluid about 3 times a day. So the choroid plexuses produce it. It circulates through all the ventricles in the subarachnoid space, and it drains into the arachnoid villi. And you can see the arachnoid villi here. If you have a problem where you have a blockage and the cerebral spinal fluid can't go through or can't be reabsorbed through the arachnoid villi, that can result in hydrocephalus. And that results in an abnormal accumulation of cerebral spinal fluid in the brain. And if you insert a shunt and drain the fluid down into the ventricles, from the ventricles into a vein in the neck, then usually you can alleviate the problem. Now, the brain receives 15% of the blood supply of the body, so 750 milliliters per minute, and it consumes about 20% of the body's total oxygen and glucose, the glucose that's traveling through the bloodstream. The brain requires a constant supply of oxygen and glucose, but there is a protective mechanism in place that prevents the brain from being affected by dangerous substances that could possibly injure this very delicate organ. So you have something called the blood-brain barrier. Now, the blood-brain barrier is made up of the tight capillary cells. The capillaries are made up of simple squamous epithelium, and they're joined very tightly together. Then they have a supportive basal lamina, and then the bulbous feet of the astrocytes form an extra barrier that helps to protect the brain. And the brain has probably one of the most protective barriers to substances getting in than any place else in the body, and that is the blood-brain barrier. So it blocks out a lot of toxic substances, but it is permeable to water, to glucose, lipid-soluble substances, oxygen, carbon dioxide, and then some other things that could possibly injure the brain: alcohol, some drugs, caffeine, anesthetics, nicotine. All those things are permeable through the blood-brain barrier. So some homeostatic imbalances of the brain, some diseases. Alzheimer's is, you see that quite often. This is a common degenerative brain disorder, and it's a progressive disorder that results in dementia. And as it progresses, you see the condition deteriorate as the person moves through the different stages of the disease. But some of the characteristics and manifestations: memory loss, short attention span, disorientation, eventual language loss, irritable, moody, confused, hallucinations. And part of the problem is the development of these amyloid plaques that will form in and around the neurons and interfere with the electrical signaling between the neurons, and eventually the brain shrinks over time. Parkinson's is a degeneration of the dopamine-releasing neurons of the substantia nigra, and we talked a little bit about this earlier. Basal nuclei are deprived of dopamine, they become overactive, and there's tremors at rest. Usually it's a unilateral pill rolling motion that you see in the beginning, and then it progresses, and eventually they lose facial expression. They have that masked appearance, stooped over, no arm swing, shuffling gait, and it continues as the disease progresses. Treatment with L-DOPA, deep brain stimulation gene therapy. Research into stem cell transplants is promising. There's a dementia associated with Parkinson's called Lewy body dementia, although not everybody has it. So there is a continuum, and you will find that the symptoms and the manifestations of the disease varies from person to person. Huntington's is caused by a dominant gene defect, and it's also called a delayed action gene. This is a fatal condition, and the person usually doesn't demonstrate or manifest any of these symptoms until they're in their 30s or 40s, and by that time most people have had their children. And because it's a dominant gene, that means that 50% of your children will have it. But it leads to a degeneration of the basal nuclei and cerebral cortex because of the accumulation of a protein called Huntington. Some of the things you will see is in the initial symptoms, you will see these wild flapping, jerky motions, uncontrollable, and then later very marked mental deterioration leading to severe psychosis, and eventually the person becomes somebody that is unlike you've ever seen before. So this person becomes very, very psychotic, very violent, and usually has to be institutionalized as the disease progresses. The life expectancy is 3 to 5 years once it's diagnosed, and there's very little treatment for this disease at this point in time. So now we move to the spinal cord, which is the other part of the central nervous system, It's about 42 centimeters in length or 17 inches long, 3/4 of an inch thick, and it provides a two-way conduction pathway to and from the brain. Major reflexes are initiated and carried out through the spinal cord. It has the same coverings that you would find in the brain. You have the same 3 layers of meninges. Above the dura mater is a space that's filled with fat and blood vessels. The fat acts as a cushion as the spine is going through the vertebrae. And the termination point is the conus medullaris. And there are 31 pairs of spinal nerves that attach to the cord by paired roots. And they exit out by the intervertebral foramina, which is the little spaces in between your vertebrae. So here's a look at the spinal cord. There's your termination point, your conus medullaris, and after that the solid cord spreads out into fibers, and these fibers are called the cauda equina, which means horse's tail. There are enlargements where nerves exit from the cord that serve the arms and the legs. So the cervical enlargement for the arms, the lumbar enlargement, nerves go out of that region that serve the legs. And here's some of your major divisions of spinal nerves, which we'll talk about in chapter 13. Cervical nerves, thoracic spinal nerves, and lumbar spinal nerves. Here's a cross-section, and it's showing you that epidural space where the fat is, your 3 layers of meninges, your subarachnoid space where the cerebral spinal fluid flows, and then you see here a cross-section of the spinal cord. And just as a review, remember, white matter's on the outside, gray matter's on the inside on the spinal cord. In the brain, it's reversed. Gray matter on the outside, white matter on the inside. So this is a nice picture that shows you sensory input. Okay, those are your unipolar neurons with the nerve cell body in the middle. And you can see the word ganglion here. A ganglion is defined as a collection of nerve cell bodies, and because your nerve cell bodies are outside the central nervous system, that is a ganglion. And then you have your ventral root, which is your motor. So motor, multipolar neurons going out. Remember we said sensory was afferent, motor, efferent. They come together here. And then your gray matter is divided into regions. Now, the ones in the back are called the dorsal horns. Okay, these are dorsal horns. The ventral is called these are your motor neurons, and this is the ventral horn. Sometimes they'll call these posterior and anterior horns too. So you may see both terminology used. Dorsal is for incoming sensory information. Ventral root, motor output. So if you have damage to the ventral part of this horn, it's going to affect your ability to move something. If you have damage to the dorsal region, it's going to affect your ability to feel something. Okay, so injuries in the spinal cord can give you a good idea where they are, can tell you what's going to happen to that person. Now these areas here give you an idea of what's coming in and what's going out. Remember I said the dorsal horn, sensory input, and then the ventral horn, motor output. So this blue part here, this is somatic sensory. So coming from your skin, coming from receptors, touch receptors, pain receptors in your skin. Interneurons in the green region come from your visceral organs. So you have feelings in your organs, and those come in here. Now visceral motor, those are motor pathways that go out to your internal organs. You do not control those because those are autonomic. So those are out of your control. But the somatic motor neurons in this little pinky red looking area, this goes out to your skeletal muscles. So these are the ones you do control. So these are the regions within the gray matter and what areas they control. Sensory and motor. Now, neuronal pathway generalizations. Major spinal tracts are part of a multi-neuron pathway, and they cross over to the other side. Remember that crossing over occurs in the medulla oblongata. And the relay, either ascending or descending, is part of a 2 or 3 neuron chain. Now, your ascending pathway is 3, your descending pathway is 2, and I'll show you. So, this is the ascending pathway. So your receptor, say touch receptor in your finger, okay? You feel something, it goes through the first order neuron, which would be a spinal nerve. That is going to connect to the spinal cord, where you have spinal thalamic tracts. Now, the tracts usually tell you where they're coming from and where they're going to. So this originates in the spinal cord, and it goes to the thalamus. That's your second order neuron. The thalamus is your relay station, and it relays the information by way of a third order neuron to the postcentral cortex. So the cerebral cortex, and this is going to be your postcentral gyrus, remember, because that's where your sensory input comes into, and that's in the parietal lobe. So your ascending pathway has 1, 2, 3 neurons in the chain. Descending pathway comes from the cerebral cortex, and that comes from the precentral gyrus, and this is in the frontal lobe. So if you want to move, say, your finger, that area of the cerebral cortex makes the intent, and it goes by way of a corticospinal tract. Those are your pyramids, pyramidal tracts. That's your first order neuron. Starts from the cortex, goes down the spinal cord, and then it's going to connect to your second order neuron, which is a spinal nerve, and that spinal nerve takes the information out to the effector, whatever muscle it is that you need to move. So your descending pathway is 1, 2 neurons. Okay. Since there are 2 neurons, there's 2 types of paralysis. You can either have spastic paralysis or you can have flaccid paralysis. If you have spastic paralysis, this part of the pathway is affected. If you have flaccid paralysis, this part is affected. Because you see, if this neuron is still affected and is connected to the muscle and the pathway is complete, you can have reflex action through the spinal cord, and you can have spastic paralysis, and you can have uncontrolled contractions of that muscle. If the nerve to the muscle itself is cut or damaged, then that is flaccid paralysis. So let's just elaborate on this a little bit more. Flaccid paralysis, severe damage to the ventral root or ventral horn cells. Nerves do not reach the muscles. There's no voluntary or involuntary control of the muscles, and muscles atrophy. Spastic paralysis, damage to the upper motor neurons of the primary motor cortex. Spinal neurons remain intact. Muscles are stimulated by reflex activity. No voluntary control of muscles. Muscles often shorten permanently. So, spinal cord trauma can be very devastating. A transection or a cross-sectioning of any spinal cord at any level can result in total motor and sensory loss in regions inferior to the cut. Paraplegia, loss of lower limb function, transection between T1 and L1. Quadriplegia, loss of all limb function, transection in cervical region. And spinal shock is a transient period of functional loss causal to the lesion, usually some traumatic impact type of an injury, but the person does recover. Polio, destruction of ventral horn motor neurons by poliovirus. Remember, the ventral horn is where motor output originates. Muscles will atrophy, and death may occur from paralysis of respiratory muscles or cardiac arrest. And survivors of polio usually develop post-polio syndrome many years after from neuron loss that occurs. Amyotrophic lateral sclerosis, ALS, or Lou Gehrig's disease, is destruction of the ventral horn neurons and fibers of the pyramidal tracts. Again, ventral horn, so motor function, and eventually respiratory muscles are affected, and that's what causes the person's demise. Death typically occurs in 5 years, and the one exception that we saw was Stephen Hawking, who was diagnosed in his early 20s and lived into his 70s, so over 50 years with the disease, which is highly unusual. Some of the theories associated with it: glutamate, excitotoxicity, environmental factors, There is very little out there to treat it. They're working on drugs, but not a good prognosis. So that is it for chapter 12.